It's a real honor to be here. I'm here thanks to Amber, who had reached out, I think it was 2 years ago, and she and Caddy welcomed me to the community. And I've learned so much, and I'm very, very happy to be here. Thank you. When Amber reached out, we tried to think about what my lab could do in a way that would be complementary to what the community does. I'm not a physician, I'm just a basic scientist, and our bread and butter is electrophysiology. So what my lab does at the University of California in San Francisco is to understand where the seizures originate in the brain and which cell types and connections are sufficient to target in order to disrupt seizures without side effects. So, the work I'm going to talk about was done by two amazing postdocs, Julia Voskobenik and Britta Lindquist, who is a physician scientist who treats patients with epilepsy at UCSF. And unfortunately, they're not here. Julia was supposed to give this talk, but she's on maternity leave, so I'm giving the talk for her. The questions that we're going to answer, as I said, we are mainly interested in which brain regions matter. Because in cases you have a loss of function, like in the case that we discussed today, right? With the loss of function of GAT1, the thing is that many cells in the brain and many brain regions can do a wonderful job to compensate for this deficit by upregulating other GABA transporters. GAT3 was mentioned. So what my lab does is to find the hotspots in the brain that are more vulnerable to the loss of GAT1 that are not able to compensate. So we are mapping the brain to find those regions, and we are using optogenetic approaches to target those regions to understand which connections are vulnerable and can be targeted to stop seizures. Before I show you which brain regions are the most involved in seizures, I'm going to show you the basic characterization of the mouse model, and many of these results have been already shown by others. But bear with me, I'm going to try to go through this fast. So in our hands, does the S295 mutation affect mouse survival? Do the S295L mice recapitulate epileptic activity? Does this mutation drive mouse behavioral abnormalities? Do behavioral abnormalities correlate with epileptic activity, or are these two separate things? Are specific brain regions more vulnerable to epileptic activity in the SLC6A1 associated disorder? So these are the questions that we are trying to address. So first of all, does this mutation affect the mouse survival? We are not going to go into details because we're running very late, but the brief answer is no. The S295L heterozygous mice have a normal embryonic and early postnatal survival, unlike the GAT1 loss of function mice. Second question, do these mice have epileptic activity? So there have been several talks today by several labs, and the answer is yes. And these mice actually have mainly spike in wave discharges, although it's not the only phenotype. But these are associated with behavioral freezing. We call it loss of consciousness. Of course, consciousness is very hard to measure in mice, so we're going to call this behavioral arrest. And you have seen this earlier. So the EEG of this mice shows frequent spike in wave discharges here in white, associated with a reduced muscle activity that you see on top in red. And the phenotype that we are seeing, the absence seizure phenotype in this knock-in heterozygous mice, is pretty similar to the one that we see in the loss of function heterozygous mouse. However, the phenotype in the knock-in mice here on top is more severe in that they have more seizures. So these mice have hundreds of seizures a day, whereas the heterozygous, just simple loss of function mice have, in general, less seizures. The epileptic spikes are enhanced by the GAT1 antagonist and reduced with ethosuximide. So if we treat even a normal mouse with something that blocks the GAT1 function, the normal mouse develops seizures that can be then blocked by ethosuximide, which is an anti-absence drug. Same in the knock-in mice. If we block GAT1 even further, then it exacerbates the loss of function, it exacerbates seizures. They can be blocked with ethosuximide. And this effect does not depend on the time of day. So we did this during the day or night, and we see the same result. A very similar result in the heterozygous loss of function mice. So does this mutation drive behavioral abnormalities? So many other labs are studying behavioral abnormalities, so our goal is really not to duplicate the work that is done. Our goal is to correlate whether the epileptic activity that we are seeing, for example, in certain deep brain regions like the thalamus, whether those are correlate with specific behavioral abnormalities, right? So we already know from the work that was really well done by other labs what behavioral abnormalities are observed in the mice, but which ones do correlate with the abnormalities in deep brain regions? That's the question we're trying to address in the mice that are chronically recorded longitudinally using multi-site physiology that I'm gonna show you in a few minutes. So, of course, there are lots of different behaviors that we can measure in the mouse. Epilepsy is, of course, much more complicated than just seizures. And we're not going to go through all types of behaviors that are measured in the mice we are recording, but let's go over the main ones. So the neurobehavioral domains that we assessed are here on the left. The behavioral assays that we used are listed in the middle here, and on the right, Yulia prepared this nice diagrams of the behavioral setup that was used. So for example, in this particular case, we used an open field. This is a square, kind of a big room for the mouse, where it's gonna move normally. And this will allow us to quantify its locomotion. In general, this is also used for testing anxiety, even though it's more complicated. Then we measure the grip test because, as you heard from clinicians and basic scientists today, one of the problems for SLC6A1 patients is hypotonia and muscle problems. So the grip test measures the muscle strength. And the elevated plus maze also relates to locomotion and anxiety. Like dark box, same thing, anxiety, locomotion. And the white maze measures working memory. And the three-chamber sociability was used to measure the sociability of mice. Tube test was also measured, done to measure sociability. So the heterozygous mice exhibit a sex-dependent decrease in locomotion and muscle tone. I'm showing you the most representative results that we found later to be correlated with epileptic activities. So here, the white traces show the movement of the mouse. I think movement measured during 15 minutes, right? So the white trace will show where the mouse went. So this is how the normal mice behave, females and males. And this is how our knock-in heterozygous mice behave. So you see a dramatic reduction in locomotion that was interestingly only observed in females in our hands. And this is just a quantification showing that females do show reduced locomotion, but not male mice. And again, this is in heterozygous mice. We don't work on homozygous mice. The heterozygous mice also show a reduced muscle tone. So here we use a grip strength, so the mouse will touch something and will try to pull. So we're measuring how strong the muscle is, and that was reduced specifically in females as well. So very brief summary, no differences detected in the Elevate Plus maze, light dark box, three-chamber sociability, and the tube test. But what do these behavioral abnormalities mean? Do they really correlate with the behavioral deficit, with the seizures we are seeing or not? And one of the answers, we're still quantifying certain behaviors, but one of the answers here is yes. So here we measure the nesting behavior. So if you give a piece of tissue to mice, they tend to prepare a nest. It's called the maintenance behavior in mice. And the mice that had more spikes here tended to be less efficient in producing nests. So I think it's really important to correlate electrophysiology with behavior in the same mice, because when we compared whether this heterozygous mice actually had a nesting deficit, the answer was no, because it's very variable, right? And it probably depends on the time of the day, whether the mouse was having seizures that day or not. But if you have the measurement of epileptic activity in the same mouse, right, you can see whether there is any correlation between spiking and the type of behavior. So that's what we're trying to do. And then the main question, which we're still trying to address in the lab, is are there specific brain regions that are more susceptible to epileptic activity in this disorder? And maybe it's not surprising, but it's the activity is coming from the thalamus. So thalamocortical bursting synchronizes with the epileptic activity detected in the cortex of awake behaving mice. So our lab is specialized in making these devices. We make them with our own hands. We make a bunch of electrodes that we implant in very different brain areas. So we usually have multiple EEG electrodes here in the motor cortex, somatosensory cortex, depth electrodes in various cortical areas, hippocampus, and the thalamus. And these are chronically implanted for many months in mice, so we can observe the longitudinal evolution of the activities in these various regions. And I'm going to show you how a normal activity looks like. This is from a heterozygous S295L mouse during non-rapid eye movement sleep. The EMG is flat because there is no muscle activity. The mouse is sleeping. And this is a normal, kind of flat, chaotic EEG in the motor cortex, the somatosensory, in the deep here layers of the cortex, normal large amplitude in the hippocampus. This is very normal during slow wave sleep. And then not much going on in the thalamus in terms of spiking. But then what was really interesting to us, and we're still trying to understand why this is happening, there was a very specific brain state, in which the seizures were the most severe. So during wakefulness, I told you that if the mouse is moving and has an absence seizure, and other speakers showed that as well, there will be behavioral freezing, right? So if there is an absence seizure, the mouse stops and freezes, and similar behaviors are observed in patients. But here we are looking at sleep. And interestingly, during sleep, so with the transition between the non-REM and the rapid eye movement sleep, this is where the seizures are like 10 times longer. Here, what you can see is that the seizures in form of spike and wave discharges, so I showed you earlier, they're about 1 to 2 seconds long during wakefulness and active behavior, but when the mice are sleeping, these are 10 times longer. So look at this. Interestingly, they are time-locked in the cortex and the thalamus, but not much activity is observed in the hippocampus. So this does not mean, of course, that the hippocampus does not participate in seizures, but it means that the seizure activities are probably the most robust in the thalamocortical system. So what we're currently trying to understand is what happens at this transition at the neuromodulator level that makes the seizure so much robust. And that I'm not a sleep expert, but we actually started studying sleep and how the seizures disrupt sleep, and why do they mainly occur during sleep. So I would be very interested to hear from clinicians what they think about this, because we found that seizures occur in clusters, and if you record a mouse for like 2 or 3 weeks, you'll find that one day the mouse will have 30 seizures in a day during sleep, the next day it won't, then the third day it will again. So I'm really wondering what happens with the 4-hour EEG recordings that were mentioned for patients, right? How variable the results might be if the recordings are so short, and might we need longer recordings. Again, I'm a mouse expert, so don't listen to me, but I think that hopefully we can learn something from our mice. Okay, so what was I gonna say here? So what we're doing next, and I don't have results to show you right now because they're very preliminary, but we're trying to disrupt the network at various locations here by stimulating optogenetically with light locally in different brain areas to try to find which areas matter the most for disrupting the seizures. And we are measuring also whether that would affect the would have side effects on the behavior of the mouse. Okay, so with that okay, so here I just wanted to show you a spectrogram from a long sleep episode. Here, the non-REM sleep transforms into REM sleep. You can see this because the low frequency bands here disappear. And this is the REM sleep, and this is where the seizures emerge, and they're associated with wakefulness. So we're still trying to understand to what extent seizures are disrupting sleep, and to what extent it really affects the cognition in the mice. And hopefully I'll have some answers next year. So conclusions, the heterozygous S295L mutation does not affect survival. This mice recapitulate epileptic activity in the form of mainly spike and wave discharges. This mutation drives sex-dependent reduction in locomotion and muscle tone. So I know that some of you said you did not observe sex differences, so I would be very interested to chat with you to understand what might be different in our assays. And we see an increased epileptic activity that correlates with decreased nesting in mice, and the thalamocortical bursting that synchronizes with seizures in the heterozygous mice. And pilot data suggests that seizures might be able to disrupt sleep, but as I mentioned earlier, future directions will determine the extent of this disruption. A huge thank you to Amber and Maxwell. This work would have never been done without Amber's phone call. Somehow she found me and we bonded immediately. And I'd like to thank my two amazing postdocs, and Katty Kang and Stephen Gray. I've learned a lot from them. They're experts in this field, and they kind of guided me on this route of what we could do in my lab that would be complementary to what the field does. Thank you so much.